Hello, sir. Dr. Rohit here, sir, from medical team Sipla.
Right. How are you? How are you? How are
I'm
you?
good, sir. So I hope you are doing well.
Perfectly fine. Pleasure.
Yes.
Hi everyone. Welcome to everyone. We are live with our webinar. Allow me to set up the context, introduce everyone. So, welcome to all the speakers and all the attendees for this inaugural episode of our series, that is Amplification, Reimagining Diagnostics, which is brought to you by Cipla Diagnostics and BOH. During COVID-19 pandemic, while Cipla has worked relentlessly by providing essential medicines required for saving life of critically ill patients, they also realized that Cipla can also contribute more by providing robust and affordable COVID-19 testing solutions and joined the battle in diagnostic space as well. Cipla Diagnostics' vision is to provide most unique and affordable diagnostic solution in India. With that, I'll now just request Dr. Rohit from the Medical Affairs team of Cipla to please take it over from here. Dr. Rohit, over to you.
Thank you, Khushbu. So a very good afternoon to all. Myself, Dr. Rohit Malabde from medical team Sipla. So I welcome you all on behalf of Cipla for this amplification series. So today's objective of the meeting is to understand the different variants of the COVID and also to understand their implication on COVID diagnostic. For this meeting, we have esteemed faculty with us. Today's session will be moderated by Dr. Ravi Gaur, who is a Principal Advisor, Spice Health, CII, Co-Chair, Health Committee, Delhi, and also Director and Chair, Medical Advisory Committee at Onquest Labs, New Delhi. Along with him, we have two panelists, Dr. Arjun Dang, who is a CEO, Dr. Dang Lab, New Delhi, and Dr. Bansidhar Tarai, who is a Head Microbiology and Infection Control from Max Super Speciality Hospital, New Delhi. I welcome all the faculty members for this session and would like to hand over session to Dr. Ravi Gaur, sir. Over to you, sir.
Good afternoon, everybody, and welcome to today's session, which is conducted by on the Voice of Healthcare platform and well supported by Cipla Diagnostics. I think thank you so much, Cipla and Voice of Healthcare, to raising a very, very pertinent question, which is now in everybody's mind, like, what is going to be the next shape of COVID? How is it going to look like? And last almost, we are almost in the touching 2 years since the first COVID, the virus was born way back in November 19 in China. And since that time, I think this the infant which was born somewhere in 19 November is now become almost like 2 years old. And that's where I think as the virus has grown from almost like 0 year of age to almost 2 years, and there has been rapid changes in the various structure of the viruses, and that's where I think it's like it's creating a little worry in everybody's mind, because we are used to fighting COVID, version 1, COVID version 2, then COVID version 3, then the 4, and we don't know how many more COVID version exists out there and what more would come. And that's a very, very disturbing factor problem. But in any case, I think we have, as of now, it appears to be that we have really, the diagnostics and the healthcare industry, scientists and researchers, basically all of them put together, have taken you can say that the control and command and control. They are actually in control of what is going to be the going ahead the shape or version of the virus. So the thing that today, the topic what we are really talking today about is that mutant variants of concern, that impact on COVID-19 testing. I think more relevant. I think this can't be the more relevant time today, because I think in the recent 2 years, probably I'll say, or 1.5 years for that matter, more precise, the diagnostics has come to the forefront. I think they have done a wonderful job. Probably without diagnostics, especially we would not have been able to, I think we would never have able to handle this COVID pandemic. They are betting from the front and absolutely taking the brunt on the face. And the way the diagnostic has shown the credibility and credentials and strength, because today we can say that nowhere happened in the world, because today India has almost 3,000 plus COVID testing labs, and most of them are ICMR and enable approved. Even all of them, for that, mandatory by law. This has not happened in the world anywhere. So diagnostic is definitely like the leading the war, leading the show, and I'm sure this is going to be the future as we move forward again. But then when the diagnostic is at the forefront now, and the what we are diagnosing is a virus. Probably we started with the version 1, which is now, of course, we have alpha, beta, delta plus. All these numbers have been given. So we started with number 1. Probably we never thought of number 2, probably number 3, number 4. So it's definitely going to be a challenge for all of us. How do we address these changing versions, or what we call as the COVID variants and various variants as we move forward? Because our technologies and everything has been developed probably from the first version, but then upgrade to second version. By the time we update our technology, I think the virus updates themselves and come out with the newer version. I think there's a struggle, constant fight. Who's going to win the war? I'm sure diagnostics will win the war. And now with this, I think background, I will have a very strong panel today. In fact, 2 of the leading people who have been from the diagnostic industry, who has been, contributed tremendously to the war against COVID, and given a lot of solutions to not only from the service delivery, but the testing, the type of testing, the technology, understanding the virus. So I'd like to welcome my panelists today. One of the panelists we have young Dr. Arjun Dang. Welcome, Arjun. And Arjun is, I think, is not a new face to the industry. He's, you can say CEO of Dr. Dang's Lab, New Delhi. Dr. Dang's Lab is one of the, most reputed name out here, and his father actually has set up a very high level global standard. So I'm sure Arjun is taking forward that opportunity. During the COVID, I think one of the most interesting things was the biggest, the fear was how do we do the COVID collection properly. I think one of the most innovative thing Arjun started, one of the first one was a drive-through kind of a thing. And that's, of course, become a norm across the country and maybe globally for that matter. Well, Arjun, welcome to the panel. I look forward to hearing from you as we move forward.
Thank you.
Then I have with me Dr. Bansidhar Tarai. Welcome, Dr. Bansidhar. And Dr. Bansidhar is a head microbiology, head microbiology, infectious control, Max Super Specialty Hospital. And all of you know, Max Hospital Group has actually, had probably had faced the best of the technologies, best of the testing, the workload kind of a thing, because this was a hospital front. So on the one side, we have a diagnostics person, one person. On the side, we have leading microbiologists who's highly skilled microbiologists and want to really know how they were facing, because what is the from the structure of the virus onwards up to how hospital is getting ready, because for the COVID testing as we move forward. Welcome, Bansidhar. I think I look forward to see hear more and learn from both of you here, and I think I'm sure the audience who are in attendance will learn more about it. So I will not move. I think I'll stop here. I'll probably move on with the first question to Arjun. I think Arjun. I think I will just just in brief. We have almost like 50 minutes with us. There will be some couple of questions out there. We have something which is like first question to you is, can you just take us through the emergence of SARS-CoV-2 virus and brief overview, where we started and where we are, a journey about it. And during the journey, I think probably you can cover something, let us know what kind of challenges you're facing in the lab, especially from the diagnostic. A lot of technologies have evolved. We have today, it started with RT-PCR, then TrueNAc, then we have CBNAc, then we have the molecular other assays, the LAMP technology, we have Acula, we have ID now. Parallelly, somebody started, they already have rapid antigen, the antibodies. And of course, the people are talking about in some countries electronics, like breath kind of a thing, or a sweat sample. Like, just can you probably address what's really happening in the diagnostic industry, how we started, and where we are? Arjun, please.
Thank you. Thank you. Thank you, Gaur sir. Thank you for a very, very kind introduction. And a big thanks to VOH and also to CIPLA for having me on this very, very prestigious forum. Dr. Bansidhar, it's an absolute pleasure to be discussing this panel with you and sharing this panel with you. So Gaur sir, as per your question, the first one was the entire journey of the SARS-CoV-2. And like all of us know about the SARS-CoV-2 and when it started in 2019 in December, and first it emerged from China. Over this entire time duration, we've had the virus that has infected millions of people around the world. And also, there has been an evolution when we look at the structure of the virus itself. Now, when we talk about any virus for that matter of fact, may it be HIV, may it be a swine flu H1N1, or any other virus, the virus itself acts in a way over time so that it can infect the maximum number of people. Now, when we look at the population from a bird's eye point of view, the virus obviously wants to infect the maximum people and be very effective and transmissibility to be high. So as the people get more and more immune or get more protected to the virus, or in other words, get stronger in evading the virus itself acknowledges that and tries to mutate on a regular basis. And this is what we call as the evolution of the virus. Also, apart from this, it is very important to understand, and I'm trying to break it up into as simple as possible, is that when a virus goes from one person to another and infects individuals subsequently, the RNA itself replicates in the human body. This replication is done by a particular enzyme, and when this replication happens, more and more chances of an error occurring while replication is what is noticed. In the entire RNA sequence and the various nucleotides that are there, these nucleotides can change and mutate from an infection that is passing on from one human to another. Now, like you also said, Dr. Gaur, that there have been multiple mutants that have been covered throughout the evolutions of SARS-CoV-2. We have the alpha variant, that is the UK one, and then the beta that came from South Africa, and then gamma, that was the Brazilian one, and now the delta and the delta plus that we've seen that caused the second wave surge in India. So obviously these different mutations that have happened over a period of time and very, very good efforts of sequencing is the reason that have brought forth these different mutants. Now, what I would actually like to highlight here is that it's not too difficult actually to sequence these samples if you have the right kind of technology with you, but actually to make sense of these mutants and the entire practice and the entire effort of getting to know these mutants better is not from a theoretical point of view, but it is for the benefit of the larger population. Now, what I mean by that is like for example, you will see that the coronavirus, and even if you refer to literature, does show mutation, but that mutation is a slow mutation. So in some literature that I was reading, it said that it mutates about 2 times every month. That is much slower than the regular swine flu or the H1N1 also. But it's very important to document these mutations and basically understand 3 things about any kind of mutation. So first is the transmissibility. That is what is the probability of it transmitting a person, and then what is the severity once it infects a person. And the third one is the evasion, or the way it avoids any kind of therapeutics, the antibodies that we have for treatment, or even the vaccines. So all in all, the entire evolution of the SARS-CoV-2 virus has taken us to various mutations. Some of these mutations being more transmissible, some of these mutations having more severity in the person who is infected. But all in all, the scientists all over the world are doing this constant exercise in understanding the mutations, so that not only the vaccines, the therapeutics, but also on a broader base, the government and the regulatory bodies can preempt and predict the next surge and take appropriate actions. Now, coming to Dr. Gaur's second question about the challenges in the lab, you'll see a smile on my face. I'm sure Dr. Bansidhar also agrees that the past few years, I think, have seemed like a decade itself. And there have been multiple challenges, obviously, because the ever evolving science behind the virus itself has been changing on a daily or a weekly basis. So, lab experts and labs such as ours that deal with high volumes need to keep ourselves abreast and updated with any kind of change, not only with the science behind the virus, but also the technology behind the kits, and also about the best practices that we see all over the world, so that we can give an accurate report and in the essence of time as well. Now, like sir was saying, there are multiple kind of testing modalities, methods, kits available. So the gold standard, obviously, like all of us already know, is the RT-PCR test, and the RT-PCR test itself is available over hundreds of kits that are validated and approved by ICMR now, and they're available on the government websites as well. Again, these kits come with various advantages, various differences, also varying turnaround times. And also, what's very important to understand is the difference and benefits and disadvantages of a two-gene test versus a three-gene test. Now, when we talk about the targeted genes in any particular test, it basically means what are the particular genes that we are trying to detect in that particular sample, and that resembles the SARS-CoV-2. Now you have different kits and you have different genes within these kits, but broadly these genes can be different also. But a very common practice among the manufacturers is to have a particular set of screening genes and a confirmatory gene. And only when both of these come as positive, or we see both of these amplifying on the graph, then we report it out as positive. And it's very important that we always look for the one with three genes, again, because of two or three reasons. The first one being the interpretation or the judgment of the result becomes easier as the number of genes increase. The second, and more importantly and more relevant to today's session also, is that now a majority of the mutations that have been detected have been seen to also affect the spike protein part. So any three gene kit that is there reduces the error or reduces the chances of a false negative test result. And obviously, apart from this, the three gene again will also tell us that, okay, is there a mutation possibility in this particular sample? So keeping this in mind, I think it's very important for us to choose the right kind of kit, understand the science behind it, and also while reporting it, even explain it to the physicians and the doctors and the pulmonologists, also exactly what testing is being done, and more importantly, the positive samples again to go to the appropriate healthcare and diagnostic bodies to do the sequencing until it is obviously available for the private sector. Apart from this, also, I'll just quickly touch up upon the various other methodologies that are also widely prevalent in India. And one of these is cartridge-based nucleic acid amplification testing, also INAT that is offered by Abbott ID, CBNAT more popular by Cepheid, that's Gene Expert. And we've also been using this for quite a few months now. And the major advantage that these kits again provide is a very quick turnaround time, especially when you have people who are admitted in the ICU or in the hospital or have any serious symptoms. These tests can actually give a turnaround time of less than an hour. Now, what it does is, obviously, your reflex question, especially from the non-medical community, might be then a rapid antigen test gives you a result in about 15 minutes. But in this case, when you're getting a result within a few minutes with a CBNAT or an INAT, you have to understand that it is a sensitivity of this particular test is as high as an RT-PCR, that is the best that it can get, versus a rapid antigen that is a much lower sensitivity. And the second thing, again, for everyone to understand is that these CBNATs and INATs are actually capable of picking up much lower a viral load compared to the regular RT-PCR. Now, what I mean by that is that a usual RT-PCR test that we do as per the Indian government's guidelines and the ICMR's recommendations are supposed to be run for 35 cycles. That's why a CT value, when you see in a report, it will be a maximum of 35. But with these CB-NATs and I-NATs, we actually run these cycles up to 40. So any kind of a very high CT value between 35 to 40 that might be reported as negative on an RT-PCR can show a mild positive on the CB-NAT and can also show the beginning of an infection or probably the end of an infection as well. So again, it is very important for all of us to understand and decide the right test at the right time. Now, as I explore the world and the entire globe in the various kind of testings that are available, I come across very, very interesting modalities. And it's actually going to be super exciting if these come to India. Again, one of these that is already ICMR approved and the most relevant right now is the Saline Gargle test. And this has been approved and also produced in India. And I think this is a great step towards making the sample collection process actually easier. In a country like India, where there is such huge demand, and I still remember during the second wave when there were so many requests for tests and even labs were stretched to their limits, a device or a way of testing that makes it easier for the patient to collect their sample, may it be a saline gargle wash, may it be just a saliva sample that is being used widely in the United States also. And when patients can self-collect their samples and then lab doesn't have to deploy technicians or waste PPE or waste time or logistics to get the sample, I think that will be a major game changer when it comes to turnaround time and the efficiency of labs as well.
Thank you, Arjun. I think for giving a very elaborate, the brief of COVID, where we started probably and what we are today. We're talking about when we had so difficulty in collecting the samples and probably nasal and the oropharyngeal swabs were taken. But today you're just talking about gargling and just and bring it back. I think that's an interesting journey the diagnostics has covered. Coming, I think I'll come back to you with some very important questions, but you have raised here, because maybe I think I'll talk to you after speak to Bansidhar. What is going to be the new lab look like? Because that's something very important. Number one, are we heading for you have a separate room where the patient walks in and a collection room become a gargle collection room, maybe something like that. Or maybe something like, breath analyzer is going to be paired. We really don't know. I think one thing which I've realized is during the COVID time, all these breath analyzers for alcohol testing was off-road. I think some people had a good time, but I think more important was are we heading for it? I think so. Probably look forward. What is going to be a labs look like, or the collection area look like? We'll come back to you later. I think in the second part. Before that, I'd like to move on to Dr. Bansidhar. Dr. Bansidhar, I think you have been, the integral part of the designing of the lab testing kind of kits, what to be used, and what Arjun has mentioned about, 1 gene, 2 gene, and 3 genes or multi-gene kind of a thing testing. So I think I like to probably what's your view, and are the current testing kits which are available, are they able to pick up variants? Are they able to pick up Delta plus? What is the shortfall? What is the pitfall? And going forward, the various technologies. Once on the one side we have a conventional RT-PCR, other side we are talking about the TrueNAc and the CB-NAc, and other side we have LAMP now and ID now. So in these, I think with this technology, will there be a difference in picking up the various variants? And if there is, how are we going to address it, and what is going to be the challenge now? Because something we will be missing out some cases probably because we might screen. And that's one question, very important question, which is being asked by each and everybody in every clinician's mind and every patient's mind. Then what is the, probably you feel that you can say that how long and how far this mutation is going to go? But that's something which is generally viruses, as I understand, but you are an expert into that field, because they probably mutate, and finally they get mutated and they're kind of a thing. When do we see that stage when the virus dying is because of the mutation and dying of its own death? Will that be feasible? Will that be possible? With this, hand over back to Bansidhar, please. Your take on that, and what do you really want to add on to much more to what Arjun has said?
Right, sir. So first of all, I'll thank the Cipla and the Voice of Healthcare for inviting me for such a great platform here, the webinar. And first of all, this is my first meeting to Dr. Gaur sir, and I heard a lot of things about Arjun Dang because his father, I have he's a teacher. Like we both are from PGI. I have heard a lot about Gaur sir, but first time I'm seeing you, and that is on the webinar. And Dr. Arjun has pointedly, we have done so much tests. So particularly my lab has done 6.5 lakhs RT-PCR till now. So that is a huge amount of PCR testing and other testing which we have done, and we have a very good experience in the last 2 years, 1.5 years. We have as a laboratory, as a doctor, we have expanded like anything because of this opportunity to work against the COVID and the testing protocols. So I can remember like initially we have a BSL-3 lab, and that we have expanded another because huge load, plus in one BSL-3 lab we may have some like space constant, so we made another BSL-2 lab and another in the smart, one in Saket, one in smart. Even that we have expanded the BSL-2 lab, we have inserted another biosafety cabinet, so 3 to 4 biosafety cabinet, and we have a lot of automated extractions, almost 7-8 automated extractions, 8 to 10 real-time PCRs. So that is the expansion part, which we will discuss much in details. First, we have to, like the second question about how long these mutations and these variants will go on. So first, the variants and the mutation, basically the variants has developed in comparison to the reference strain, that is the one strain, that is HUN, the original strain, or the US HU strain, that is the first USA strain 2020. Any deviation in the sequence of the virus, that is lead to the variants, and we all know the variants of interest, variant of concerns, and the variant of the high consequences. Dr. Arjun rightly pointed out that suppose if we see the COVID, there are 2 nucleotide changes per month, and influenza there is 4 nucleotide changes per month, and HIV 8 nucleotide changes per month. So these are the low stable viruses. So we have because the COVID is so in the media phase, and that's why a lot of sequencing. I was going on literature, that is 1.3 million RNA sequence has been done, and they have prominently around 3,943 something. The most important variants they have analyzed. So there are a lot of sequencing and variants has been done, and we have not seen before. Although we have seen mutation in influenza as well as in HIV, but that time our medical fraternity or medical healthcare was not doing so much sequencing. But nowadays, the sequencing is very, very important part of like tracking of viruses. And Dr. Arjun has really highlighted the PCRs, but as like in the theory, there are 2 things. These and the practicality of testing is different things. So if you see theoretically, yes, because the most of the targets is the S1 proteins, that is so basically it is a structural and non-structural proteins. So mainly the structural, we all know these are 4 proteins: the spike protein, the nucleic acid, membrane, and envelope. Whereas we have there are many, I think 7 to 9 are non-structural proteins, but these spike proteins are very highly mutagenic things. So most of the vaccines also developed against this, and most of the kit also targeting on on these spike proteins. So definitely, but because these are a lot of variants are coming, so there is a chance of false negative results. However, whatever we come across, see basically how we will know, because the antigen antibody is these are the 2, one aspect. Another is the PCR. PCR, we have multiple targets, maybe 2 or 3. What we are doing is the 3 targets. So multiple targets have lesser chance of like avoiding the detection in the PCR system. But if you see the sequencing is done after the PCR. So whatever the PCR detects, we send the RNA to NCDCs and to the IGIBs, and they are doing the even the now we are sending the ILBS also. So there is a sequence of lab. The Indian guidelines also. They have also in the July they have in the Indian SARS-CoV-2 genomic consortium INSAC has made, where they have made out 28 labs. The satellite lab as the hub labs and the satellite labs, where there is the sequencing is being done. So, but that is being done when we do the detection PCR and we send the RNA copies to them. But randomly from the community, they have not sequenced. I don't think much data they have, which can say these are the missing in the PCR and they are able to detect in the gene sequencing. So there is not much data where you can say practically where there is which is missed in the PCR, COVID RT-PCR, and which is not able to detect in the PCR, which is not detected by PCR and which is detected by the sequencing. So this is the so that's why on the practical aspects we don't think much that the variance will be. We cannot able to detect the variance. Most of the variants, even the Delta one, the most severe one, also we are able to detect. And there are a lot of other literatures. There are alpha, and in the USA there is alpha. There are some PCR kits. I think one literature I found there are 4. FDA has given there are 4 kits test. That is Aquila SARS COVID test, then Linea COVID-19 assay, then TAC-BAT COVID-19 combo kit, and Export Express. These are the 4 kits which I got to the literature in the FDA, where they have find that these kit companies may not able to miss the COVID RT-PCR kit. But however, in a practical sense, what we have done around 6.5 lakhs COVID PCRs, we are not able to like miss anything. But in the FDA and WHO has very clearly said there is some false negative can happen because of multiple mutations happen. There are so many mutations in a single genome is happening. So all has to be related to clinically as well as the history aspect and the clinical aspects. And lastly, what your point is that how long this mutation will be going on. Probably this mutation will be going on. It's a continuous process, but I think another 6 months. What about the pandemic? Usually, what we have seen before also in 1918 and 19, the pandemic, and here the pandemic usually stay for another like 2 years, which will be covering I think December or maybe March. For us, it is March 2022. So that time the mutation will be maximum, but then the continuous mutation will be happening, which will be not bothered about that mutation because H1N mutation is still going on and we are able to detect the H1N and influenza virus. All respiratory viruses will have some kind of mutations. So I think that is, I hope that is what we are discussing. Sir, right towards you, sir.
No, it's nice. I think you elaborated pretty well about the virus, and I'm pretty hopeful. And I think the good thing you're talking about, maybe another 6 months at the max, virus is going to die his own death. I'm sure because this is not probably go off around, but it's going to be there in some form or other one. Maybe little some from pandemic to epidemic to endemic, and probably some the highly susceptible population. Probably it'd be that. I think one question for you here, because I think what even Arjun has mentioned that, have you seen like over now, you've been almost testing, I think 5-6 lakh samples you've tested, or you're almost like right from the start, you've been doing PCR testing. So, there are kits like, single gene, 2 gene or 3 gene. Do you have, have you seen the changes from there? Because that's something when we started there and now we are today, number one. So are the newer kits what you're getting is probably covering much more, or it's just a similar thing? And going forward, what going to be the structure look like? And number two is there is a need, I think definitely some extent, definitely need to that clinician fraternity of people to be aware if this kit or some kind of a thing covers 3 genes or 4 genes or 5 genes, whatever something. I think any views on that?
Right, sir. So, like pointly, you sir, what do you ask? Basically, the multiple genes or 2 genes or 3 genes or 4
Or I like
genes.
to just interrupt here. Or will you like to have, in your lab, you have 2-3 different varieties of kits because depending on whatever it is.
Yes, sir. Yes, sir. So basically, sir, why because then again coming to the theory part, because in the PCR systems we are using the primers and the probes. And if you see the probes where it is being attached, like it is attached to the probes or the primary where they attach to the RNA, if that part got mutated, then it is difficult to catch up in the RT-PCR. This is on the theory part, but basically we have, if you have a multiple targets, if some primer probes where the attachment site is where the annealing is happening, if that part is not able to catch by some primer or probes, and then will catch by another probes primers, then definitely multiple targets. CDC and WHO also has mentioned that multiple targets will have a better. This they have mentioned in the literature also because of this only. Number one. Number two, about the more than 3, like 4 or 5 targets. That is another things. Most of the times, 3 target is sufficient. Even the a lot of biofar film array also they have also 3 targets. But basically, sir, if you see the structural, non-structural genes. So if you have a combined of both structural and non-structural genes, like the ORF1, where that is of non-structural genes, or the structural, if you see the from the spike proteins or something like, if a combination of both structural and non-structural target will be there, then there will be less chance that the mutant will be missing or the variant will be missing. So I hope the companies or the company, they will target at least multiple targets and they have cover all the parts where there will be a less. I was seeing that the N region is low, more of conserved region rather than the spike proteins. Although most of the targets are the vaccine developed against because that is the immunogenic part. The spike protein is the immunogenic part. So all the antigen antibody are more of developed on the spike proteins. Whereas the PCR, you can have both the structural and the non-structural genes where there is a less mutation or prone area. That will be a better effective against the targeting the PCR.
I think probably that's an interesting talk you're talking about, because I like to go from the clinical point of view, because if there is a patient who is just like highly suspicious of COVID, and you clinical and fitting well into the clinical picture of the COVID, you do of course no test is 100% correct,
yes,
because it will be always some error. But if you do a testing with one specific kind of a technology or a kit, and you say it's negative, but clinician says positive, do you think that there's going to be a possibility, okay, this is a negative, let me try from another kit, or you want to repeat the test, something, or maybe a one kit testing, a second kit testing? Do you think the future is going to be something like that, or you have a multi-kit, multi-gene kind of a combination of all this is together? I don't know. I'm just guessing around, like probably futuristic point of view. Will the lab be gearing for that?
Yes, sir. Multiple kits, because we are reference lab, so we have a multiple kits. Sometimes, like what you said, you are also reference lab, and Dr. Dang also reference lab. We have a multiple kits. But if you see our satellite lab, they have only one kit. They have to take a kit, and that should be a good one where they can cover everything. And like what you said, clinically, we have seen many cases, sir, where clinically it was there and we missed in the RT-PCRs. Because we all know that the sensitivity of PCR is 65 to 68% PCR, and that depending on the sample type. That is, I'm talking about the nasopharyngeal or oropharyngeal sample. The sensitivity will go higher and higher if you say sputum sample than the respiratory bowel samples. That is, but it is difficult to get the bowel sample from the COVID high risk patients. So the sensitivity is higher in the lower respiratory samples. And if you see the CT scan, the sensitivity is almost 68%. So the day of testing, that is also important. The day of radiological things are important. The clinical. So yeah, what you said is very much like it is not the only PCR which is the gold standard, but you have to correlate everything. And second testing is recommended definitely if there is a strong clinical. And if there is a different kit, then that is much better, because some kit have a different target and another maybe a different target with proper sample collections. And what you said, like we have done a lot of things where if it is missing in the first case, we have another kit. But we as a reference lab, for you and Dr. Lam, we all three have reference lab. We have different kit. We can test it and recheck the things. But for a simple satellite, a simple lab, we should be select a good kit where multiple targets should be there and the good sensitivity and specificity. And overall, we can miss cases because clinical radiological is more of sensitive than the PCR test.
Okay, I think that's well said. I think I think probably I can say around like, let's be very careful on as we go forward, selection, what type of kit to be used. And it will geographies probably, because somewhere we have some variant, somewhere we have another variant. And probably that is going to be very important as we move forward. And that's where the diagnostic kit providers probably have to work out. And maybe the zone specific, because variants will come sometime later, because I mean, as of now, we have India, Delta Plus, somewhere in the South African country. So maybe some kind of a different kits here and there. I think that is a continuous process and evolve. And that's interesting, I think, for all of us to know, like we need to keep an eye on what's happening and probably creating our own data bank, probably comparing with the different kits and coming out results and coming out with almost like as perfect result as possible. Although test is 100% correct. Probably, but I think as long as we achieve 95 and 98%, that's going to be very helpful to all of us. Thank you so much. I think I move back to Arjun. Arjun, you have raised a very, I think first question I'll ask, but there are some questions in the, the question bank, the chat box for you. And one, I think, where you feel that, over the time, like you can say, you have touched the kind of the testing, or probably why these variants are important for, because what is the clinical implication, what are the challenges, and which these variants, like one variant has come. Have you seen patient coming out with the first time have a COVID and second COVID and third COVID, any difference in their presentation or anything like that? Clinically, these variants, how are they, and how do we as a diagnostic provider address them? Any views on it?
Sure. So very, very pertinent question. Now that we are almost more than a year and a half into the pandemic, and we've actually had a lot of learning, I'd like to actually answer this question in two parts. One is obviously related to RT-PCRs and the reinfections that we are seeing. And also second, the entire science behind serology testing and antibody testing, and what is the effect of the mutants on the various serology testing that we are doing. So then again, we have had dozens of cases where we've seen a reinfection, probably due to a mutant, and also when the same patient had a significant titer of antibodies as well. And obviously, in a majority of these cases, reinfections have always been very, very mild compared to the first episode where they had a serious infection. But yes, we have seen these positives on a second time as well, and that's why it's very pertinent that no one lets their guard down, even if you're vaccinated, even if you've been infected in the past. So that is the difference in presentation, where a majority have shown milder symptoms. Now, coming to the serology aspect and what mutants, the mutants of the SARS-CoV-2 have done to all the serology testing. So for this, to understand this, we'll have to, although Dr. Bansidhar has already given a very good idea about the structure, but I'll again give an overview wherein we have the spike protein, where it's divided into S1 protein and S2 protein. The S1 has various subparts in it. One of it is RBD, NTD, CTD, etc. So now you get a lot of these serology tests or antibody tests that are only targeting the RBD, that is a part of the spike protein. So in these cases, if it's a variant and we use a kit for antibody testing that's only targeting the receptor binding domain, you might have a chance of actually getting a false negative. But any of these antibody tests that actually detect the entire spike protein, or now what they call is the trimeric spike protein, That includes the S1 and the S2. And these tests have a higher probability of not giving any kind of false negatives. So again, it's very important for the general population and the physicians also to understand the exact utility of an antibody test, and also then differentiate between a spike protein antibody test and also between a neutralizing antibody test. So to understand how protected the person is, you need a neutralizing antibody test that is now being offered by various labs. And also this is a pseudo neutralizing antibody test, but gives a very good idea about the neutralizing capability and capacity of the person. So again, people when they need therapeutics and also for plasma donation, etc., this might be something that needs further attention and further research also for all of us to learn more.
Okay, I think now I think we move from PCR to antibody. That's a very important question, which is a burning question, because I think as we move forward, probably, the number of almost at some point of time, all of us going to be, we are already vaccinated, we're going to more people vaccinated. Many will have almost COVID. So antibody, which antibody to be tested, what to be tested, what point of tested, and what is the clinical relevance? That's the important point you have touched on. So I think that means that you need to be very careful again when it comes to antibody testing. What kind of whether it's a spike protein antibody, whether it's a IgG, whether it's a neutralizing antibody. I think that also plays a very important role, and labs have to gear up to keep an awareness about it and the kind of testing, because I'm sure, because going forward, this will also be one of the tests will be now, as we go forward, people become more aware and they really want to know the status of what kind of a status they have, what kind of antibodies they have. Dr. Bansi, what will you like to probably say just quickly what he said, which is preferably which kind of antibody for a general kind of a screening, and what is the specific what he mentioned? Any quick take on that?
Sir, like the antibody, what we do is that IgG antibody, and the IgM antibody is not, ICMR is not approved. So IgG, we will be discussing about the IgG antibody only. So like Dr. Arjun has rightly said, they have the spike protein is the most immunogenic part, and we are using most of the in the like company has manufactured the spike antibodies, the IgG antibodies. Now we have the trimeric antibodies. We have the new things which basically that is the subunit level, the spike 1 and spike 2 subunit levels also. There are 3 targets, the RBD as well as the CBD, and another thing, there are 3 targets are there. So basically, because they have not researched most of the other antigenic part, like the non-structural part. So that's why, because that may be some vaccine may not be giving some, like even the Covaxin part, what I'm discussing about the Covaxin things. So depending on the mutant, variant, and the antibody type, like the alpha, beta, gamma, delta, what we are discussing, some like the alpha and I think gamma is a good antibody level, like, but beta part was going through the literature. Beta strains may not give good antibody level, may escape some antibody levels. Delta, we have come across, so we are getting some antibodies. So most of the IgG antibodies are it's against the target, the S protein and spike protein, because that is the most immunogenic part. And then this, as for the variants, we may escape some of the antibodies, but what the PCR antibody are two different things. Immunogenic antigen maybe in the variant we miss, and antibody also, but in the PCR, I think overall it's good, better diagnostic kit if you compare to anti-immunological testing.
Okay, I mean that means that when it comes to antibody, spike protein is something which is going to be the norm as of now. Yes, and yes. I think as we move forward, there may not be much change, but there will be milder things as we move forward. More because we really don't know how the mutation is going to behave, what is going to mutate, and how it's going to look like. But I think we are fairly confident about it. Like this is something probably it will not go out of control now. We have taken command of the COVID now. But I think before I move on back to Arjun, I think one question has come into question back. This is something like, there's a doctor who's facing a lot of cases where N gene is coming negative, while the ORF comes positive. Really, I want to know how do they report these tests, because that's your expertise. You can actually.
Sir, engine is a non-specific, so that can be engine is a non-specific. So ORF is more specific, so definitely it will be a positive. So that has to be, if clinically it's okay, so we can give. ORF is more specific.
Perfect. And that's answered the question, I think, in the box. Now, I'm back to Arjun. I think we have, I think, another about 7-8 minutes. But I think one of the, you touched very important, the point, self-sample collection. So I will actually like to, like, really, where do you, I think question which is also in the box, right, for you is like, how far, how reliable and how sensitive and specific are these gargle-based, Saline gargle-based kind of testing are, and where are we? How far we are away? Are we going to be offering this into clinical setting and diagnostic labs and hospitals very soon, or it's just still going to be research work?
Sure. So very, very interesting times with these non-invasive or self-sampling, like we are calling it. And I think if all of us, I think, are already aware that the test kit for rapid antigen testing that has been approved by ICMR, I think the one that manufactured by my lab, and also now a couple of other kits that are available for self-testing at home with a mobile app will be the starting step for any of these self-sampling test kits to become abundantly in use. Now, obviously, there are advantages because this increases accessibility to the test and ease for the labs also to a great extent. But I think the major challenges would be, again, monitoring. By monitoring, I mean is that the main purpose of why we are testing and reporting the reports also in a timely fashion to the government is so that they can be timely tracing, tracking, isolation, treatment, etc. So when people start performing these on their own and getting results, probably in a self-test kit like a rapid antigen test kit at home, then obviously we would want everyone to be very honest and updated on a real time. But I think it would be a challenge where the person themselves is collecting and testing. Obviously, number one challenge would be awareness and education about exactly how to collect the right kind of sample, keeping in mind that the RT-PCR, like Dr. Bansidhar said, has a sensitivity of just about 70%. Obviously, the sampling, if there is any compromises on the kind of sample, the sensitivity takes a huge hit. And then that is just going to lead to more and more false negatives. So again, monitoring the test, awareness and education about how to collect the particular sample, and then timely transportation under the correct conditions to the reference lab where it's being tested. I think if together we can brainstorm and come up with solutions to combat these three issues, then I think this is a great success. I think this is the key to the lock of coronavirus testing in India, where we can probably amplify the testing to maybe tenfold, if not more, with the kind of infrastructure that we have established in the past year and a half. But obviously, a lot of education awareness will have to go into it as well. Taking the United States as a case in point, they've actually done huge number of saliva testing wherein the container or the sample kit is picked up by the person or is shipped to the particular patient's house with a brochure or a video link that explains to them exactly how the samples to be collected. They collect the sample, they pack it, and then they post it back to the lab, or the lab has a logistics team that picks it up. But obviously, so we've also done an in-house study with a very renowned academic institute in the US, and we continued this from the beginning of the second wave up till now, and it is ongoing. What we saw in our concordance study in saliva samples versus the NPOP swab, that is the usual way of collecting samples here, was that any nasopharyngeal, oropharyngeal RT-PCR test with a low CT value was correlating very well with the saliva sample. But unfortunately, any cases of nasopharyngeal, oropharyngeal sampling RT-PCRs that were giving a CT value of, say, more than 30, 32, those were coming as negative in the saliva sample. So in a broad kind of a viewpoint and an opinion, I would say that yes, this is a very, very good way ahead, especially for our country. But then again, physician and patient awareness is key. And also a lot of, we have to be very careful while actually shifting these modalities, because it can certainly be make or break.
Yeah, I think that's very, I think, relevant point, because the awareness, and that's important point to moving from different type of sample from here to there, and how to transport the sample, because those could be infective sample, right, left, and sample. If we don't know, I think that awareness and the patient, I think that's very, very important role, and that's where I think diagnostics and the probably the clinical fraternity has to play a very, very important role. Like, this is the way we need to handle. Although I'm sure as going forward, self sample collection is going to be a norm sooner or later. But I think we're a little far away from there. I think we need to really, be careful about it, and create more awareness and more safety measures. Otherwise, this might boomerang and come back again, create difficulties. Now, last question, I think, before we come up, I think Dr. Bansidhar and Arjun, both of you, when we have so much of variation, where the variants are coming up, because we have alpha, beta, gamma, delta plus. I think how do we know the mutations by sequencing, like in sequencing and signature imprinting? We don't understand that. Now, when do we see from here now? Because I think going forward, COVID is going to be there for a couple of years. I think at least more than 2 to 3 years, definitely, and maybe more longer time also. And some like maybe some in the endemic phase, or something, once in a while is going to be there for a while. So when do we see the sequencing coming into the diagnostic picture for general public in masses? When you feel that a patient comes to the hospital and you want to do, instead of an RT-PCR, you want to do you're positive, but you want to know which variant it is. Do you think this should be now introduced, the time to introduce that sequencing to the public? It should be part of the hospital offering, or part of the OPD diagnostic lab offering, at least a referral lab offering? Currently, it is just restricted to research point of view. Going forward, what should be your views? And I think we should we move ahead and take this and to the various authorities? I am sure there are these kind of questions have been raised to the authorities, and say, like, we would like to do offer sequencing. What's your views about it, Arjun and Dr. Pansal?
Sure. So I feel, sir, that sequencing is what is going to save any kind of future surges. It is absolutely the need of the hour. And I feel that, like Dr. Bansidhar said, that we've been sending our samples to the NCDCs and the other research institutes. And I would say that that was the right kind of move to start with, because for the researchers and the scientists also to understand the virus and the mutations better, it's very important that the lead is taken by these government institutes, so that on a larger base, government policies, lockdowns, predicting these surges can be done effectively. But I think how the RT-PCR diagnostic tests also slowly and steadily were given to the private sector, and the private sector did this great work holding the government's hand. I feel it is time, it is certainly time right now, when the cases are at their least, so that the government can get the private players' help, even for the sequencing purposes, so that more samples can be sequenced and we can get more data and more knowledge and be more ready for the upcoming third wave.
Okay. Dr. Bansidhar, your take.
Yeah. Sir, like what you said, like many patients also, particularly in admitted patients, they have asked me, can I know what about my sequencing results, or which variant I have? So people are asking about this, patients are asking about this. But for the diagnostic, what I feel, sir, like the surveillance is very good. Definitely what we are ongoing is going on. Where the diagnostic will be help in sequencing is that the antiviral therapy. So the variant is resistant to this antiviral, or the monoclonal antibody will be given this variant or not. When the treatment aspect is there, then sequencing definitely it will be helpful. Like suppose in oncology sequencing, if you need a targeted therapy, so that is 100% in all oncology sequencing is very, very important. But here the antiviral aspect, how we will come up, the antiviral resistance pattern, the antiviral treatment therapy, how it is coming up. If you have very good antiviral therapy will be coming in the market, definitely in that case, sequencing will be a clinician will be requiring. But as of now, the mild symptoms those are basically the contact tracing. As of not in a front line, but when a good antiviral drug will be available, in that case, I'll recommend sequencing is recommended for the diagnosis.
And I think that sequencing is going to be, we're going to see the near future sequencing becoming one of the tests. That's something I think we all should get ready. I think labs are getting ready for that.
Right.
and we should be getting it. One quick question has come in the box, Dr. Bansidhar, for you. Like, what about RT-PCR kits for CSF, synovial fluids, and body fluids? Do we have something otherwise, or we're
No,
ready?
sir.
Not yet.
No, sir. No, sir.
And we definitely
And,
like to keep in the future.
this is not recommended ICMR, and also unofficially we have done few tests because of clinical forces from the gastro people, also from the body fluids also. Unofficially we have done, but that is negative. So that is, I don't think it will come. You can see alternative method of diagnosis, but not the PCR. Maybe antigen antibody that we can think of, but not PCR.
Okay, that's something currently we are not, I think, doing that, and that's really not available. Arjun, I think one more question has come for you here quickly before we, I think 2-3 minutes more I'll take, not more than that. They're talking about, this, I have just read it out, the long COVID and autoantibodies. Can you highlight on that?
So I didn't understand the question clearly, but
Question
yes,
is saying long COVID, and you actually probably answer longer COVID means the COVID duration is from 14 days to goes to 1 month, or maybe post-COVID kind of a thing, and something. You have seen something in the practice because.
So what we've seen is a lot of cases of long COVID, and long COVID is essentially defined as any COVID infection that shows symptoms more than 90 days. And in these cases, we've had people present with headaches, body aches, fatigue, difficulty sleeping, that's insomnia, etc. But so these antibodies again can be measured over a period of time and monitored as well. And obviously, like we already know, the immune response has been seen to be varying among individual to individual. And what theory says is that these antibodies should last for a few months. But in certain cases, we've also seen these antibodies last for more than 6 months as well.
Dr. Arjun, see, basically, what I feel, there are few cases of autoantibodies patients we have come across the neurological neuritis, and a lot of patients in the neurologists, they say like the post-COVID, after post-COVID, long COVID, 1 month, 2 months time, they have seen post-neurological changes, autoantibody changes. But sir, there is no significant paper on this, but they are sending a lot of autoantibody tests post-COVID things. So that maybe this is the answer they are looking for.
So I think that's going forward. The journey of COVID is going to be more interesting. I think more and more we're going to know about it. I think we think that we know a lot about it. But I think much more is to be known about COVID. And but yes, I think I can show you I also come from diagnostic background. We have experts. Bansidhar and Arjun right here. They have been right in the forefront from the day one, seeing every aspect of COVID and all variants of COVID and how they present clinically, what are the challenges, what technologies are evolving, what tests, right? Whether it's a PCR or the rapid PCR or another technology of the PCR or a kind of a thing, antigen testing or the antibody kind of a thing, and maybe the different type of samples also. I think this is a lot more to come, but I think one thing I like to definitely, because there's a question which has come in the question bank, are we ready for the third wave? Yes. I can definitely show you from, I think on behalf of the entire diagnostic here and a lot of diagnostic industry people, today we have geared up. I think they don't need to fear, no need to worry. We are ready for face the variants. We are well equipped for the variants. We are, in fact, labs, our diagnostics and hospitals are geared up to fight take up this thing very well. And that's something which is like going to be the future. And we probably much better refined kits and all things going to be out here. And we will be definitely doing something which is the required if at all we see the third wave. We hope and pray there's nothing like third or fourth wave, but there's going to be some changes as long as we have all these adopt safety precautions, still using masks, hygiene, safe distancing, and of course be careful, not like what we have. Probably we went out of the way and probably we when the first wave got over and suddenly we got, everything is over, the wave came back. I think it's time for us together to defeat it. And the diagnostic providers, we are ready, we are fully geared up, and we will definitely like to show you on that path. With this, I like anything, Arjun, you'd like to add, Bansidhar, or I think we just call it a day.
Yeah, sir, one point I want to, like the words we have discussed, but one thing that there may be cell-mediated anti-immunity, which is also working from the behind, which we have, so a lot of variants can also be effective on that only, so that is which I think that will be also, we should not see only antibody, IgG antibody. That is one. And I think another question there, someone asked about pediatric cases. Of pediatric, basically third wave, nothing. In pediatric, there may be of high transmission, right? But the protection they have much stronger than the rest of the, like the adults and the.
Arjun, anything to sum up?
Thank you, sir.
Thank you. Okay.
Thank you, sir.
Yeah, thank you both of you, and thank you Voice of Health and CIPLA here. I think very interesting discussion. We really have opened up, but I think this is come what may, whatever it is, how many reprints of COVID can come, but I'm sure the original, we know the original book, original signatures of the COVID, and I think we will be able to fight it out very well. Once you know what is the originality of anybody, we understand it very well. The diagnostic industry has understood it pretty well. And so we are going to be, although evolve, journey will continue. I think better technologies, better things are going to come, whether the kits, whether instruments or turnaround time based, because we are going to see something COVID as a screening test as we move forward, where people will be more dependent on the turnaround time. So probably labs or hospitals will be equipped with multiple platforms, because something to offer in 10 minutes, something to offer in 15 minutes, something to offer in 1 hour, or maybe in the evening. I think these combinations are going to be there. Structure will change to some extent, but testing remains the same, and we are all ready for it. Thank you so much.
Thank you, sir.
Voice of Health. Thank you, Arjun. Thank you, Bansidhar. With this, Dr. Rohit, are you there on the line? You want to add on?
Yes, sir. So everything was summed up very nicely, sir. So on behalf of CIPLA, I would like to thank Dr. Ravi sir, Dr. Bansidhar sir, and Dr. Arjun sir for the very excellent session and very informative session for all of us. Thank you, sir. And lastly, I would like to thank all the attending doctors for sparing their time and attending the session. Thank you.